Swayam Prabha. Digital India. Educated India.
Welcome back to our course Industrial Biotechnology. Today I think in the last lectures I tried to cover activated sludge process and anaerobic digestion process for the treatment of industrial effluent, and today I want to share some of our research experience that we have on biohydrogen production. We consider the hydrogen should be fuel for the future. The reason is that when hydrogen burned, it produces only water, and not only that, hydrogen has highest energy density as compared to any fuel available with us. So we want to share that kind of research activities that is going on at IIT Kharagpur, and recently we carried out 10 cubic meter pilot plant study. The research, the information on that also I am going to share with you. And finally, I will show you the video, one operational video on the operation of pilot plant, so that you can have some kind of experience how the industrially that we operate the fermentation process. So let me start with that. This is the topic that I have chosen, that is the 10 cubic meter pilot plant operation for biohydrogen production as IIT Kharagpur. And if you look at the fossil fuel, that is production and demand, it is demand keep on rising with respect to time due to the rapid industrialization and urbanization, and you will find that after 2020 around that our demand keep on rising, but the fuel production from the fossil fuel that will keep on declining. So there will be gap between the demand and the production of fuel from the fossil fuel. So naturally we shall have to find out some kind of alternative resources to fulfill the gap. So this is the fuel usage. It is very interesting. If you look at the human civilization, we started with burning of wood as a source of energy, and slowly we switch over to a particular area of what we call hydrogen and fuel cell, where we don't have any kind of pollution. In the fuel cell, we can use the hydrogen as a fuel, and it converted to electricity. So hydrogen will be converted to water here. No way it will be polluting the environment. So this, if we can do that, we can save our environment, and we can also solve our energy shortage problem. Now hydrogen, the unique energy source, the reason is that it has no greenhouse gases, no ozone depleting chemicals, no acid rain ingredients, no pollutants, clean environment for biodiversity, high utilization efficiency, conserving resources, abundant energy for economic development. Now if you look at the current technology for the production of hydrogen, there mostly it is the chemical and the electrolysis process, and these processes are, as for example, the thermochemical gasification coupled with the water with a gas shift reaction, and then first pyrolysis followed by reforming of carbohydrate fraction of bio oil, direct solar gasification, miscellaneous noble gasification process, biomass derived syn-fuel conversion, supercritical conversion of biomass, and microbial conversion of biomass. So we are here microbial conversion of biomass, because biomass is a renewable energy source. If we can we can produce some kind of useful fuel from this biomass, this will be sustainable. So now question comes, why you are interested for biological hydrogen production processes. The reasons is three, that one is what we call essentiality, another is the versatility, and another is the naturality. Now if you look at essentiality, that means that constant that we have with the conventional techniques. What is the constant we have? That is the high temperature, high pressure. and a high energy intensive process. But this is to be replaced by because biological process usually operated at ambient temperature and atmospheric pressure. So energy requirement for this process is comparatively low. And versatility is that this energy can be derived from the renewable energy source, particularly waste material, which which not only safeguard our environment, but also the waste material can be used for the generation of energy. Naturality is the near zero pollution and cost effective process due to the simple technique. Now, if you look at the hydrogen production from microbial conversion of biomass, this is the industry that produces flue gas which polluted our environment. This carbon dioxide can be fixed by carbon dioxide sequestration process by using algae. We have algal biomass. These algal biomass can be used for hydrogen production. The raw materials, different agricultural residue, different organic waste can be used for the dark fermentation process and converted here. The spent media that can be used for biophotolysis can produce hydrogen, and dark fermentation directly can produce hydrogen. And when this material can be used in the microbial fuel cell, that also produces hydrogen. So this hydrogen we can pass through the fuel cell for the generation of electricity. Here the product is water. This is how it is what you call it is a sustainable process, because we don't pollute the environment. At the same time, we produce our use our energy for fruitful purpose. Now, if you look at the roadmap of biohydrogen research at IIT Kharagpur, when we started this research work 1999, we isolate and characterize the E. coli, IIT BT08. These organisms appears to be the first hydrogen producing organism so far been reported. Then this is the organism we reported for the first time that this produces hydrogen, and then we try to justify the reason behind that by doing some kind of molecular biology work. We did the redirection of metabolic pathway for increasing the hydrogen production. Then we used the continuous hydrogen production by whole cell immobilization system, optimization of physicochemical parameters for hydrogen production. We did modeling and simulation for hydrogen production, effect of iron for fermentative process. Then we used the hybrid reactor, and then now we have successfully operated the pilot plant for the. Now this is just we are looking for the industry for the commercialization of the process. Now, if you look at the biodiversity of the biological process is like this. This can be produced dark fermentation process appears to be the best process, because we can produce hydrogen from the organic waste, and we can produce hydrogen throughout the day, because it doesn't require the presence of any kind of sunlight. But if you whenever we work with the photobiological process, it depends on the sunlight. That is another additional requirement that we have. So not only that, this problem, direct biophotolysis and indirect biophotolysis, one is with the help of cyanobacteria, another is with the help of green algae. And this we found that when we use this organism, and if you look at the history of biohydrogen production, is very interesting. That Misui, that was scientist from University of Miami, that he found out that the algae can produce hydrogen from water. And as you know, the 3/4 of the earth is water. So if we can produce hydrogen from water, we can solve our energy problem. But the problem is that when we try to grow this algae, we find that when water is hydrolyzed, it not only when water degrades, it produces hydrogen and also molecular oxygen. This molecular oxygen give the inhibitory effect to the enzymes hydrogenase, which is responsible for hydrogen production. So initially the hydrogen production started, and after some time this is inhibited. So research is going on how to safeguard this problem. But we find we don't have much of problem when you go for dark fermentation process. The one advantage is that we don't require any kind of sunlight, in addition to that, hydrogen can be produced from the waste material, and it can produce throughout the day, and it produces hydrogen at a very faster rate. And another problem that we have with photofermentation process is produces some kind of pigment. When it grows, it gives some kind of shading effect, and so light penetration is a problem. So that light energy conversion efficiency, that will decrease with respect to time. As the time proceeds, the conversion efficiency decreases to a great extent. This is the major problem that we face with the photobiological process. Now here we can see the dark fermentation, and this we had the collaboration project with the University Oslo University and Technical University of Denmark and Uppsala University, just to find out the potentiality of algae for the hydrogen production. We have come to the conclusion that this is no good for hydrogen production. And then there is another process called photofermentation process. In this photofermentation process, the photofermentative bacteria can utilize the metabolites which is produced in the dark fermentation process for the generation of hydrogen. But the problem is that, as I told you, that as the organism grows, there will be kind of shading effect, and that causes the reduction of hydrogen production to a great extent. So we find that is why the dark fermentation is most suitable for the hydrogen production. Now, if you look at the dark fermentation process, how it works, is that this is the raw materials, biomass, agricultural waste, and we have organic waste. This is this is biomass. We mean the activated sludge that we get from the activated sludge plant, that is the biomass, and we can agricultural waste. We have several. Then organic waste. This is first it should undergo pre-treatment. After the pre-treatment, we take it in the fermenters, where the dark fermentation take place, and then we get the gas. One way we get hydrogen, another way we get carbon dioxide. Now, dark fermentation usually carried out by two groups of microflora. One is called mesophile, another is thermophiles. Mesophiles, we have two categories. One is obligate anaerobe, and this is facultative anaerobe. Now, facultative anaerobe is easy to handle. The reason is that this can tolerate little bit of dissolved oxygen concentration, but obligate anaerobe doesn't tolerate. They cannot tolerate that the little bit of oxygen concentration. The organism will be killed in presence of oxygen. So this is the problem of creating the anaerobic environment, complete anaerobic environment. But once we create that, then the organism grow very nicely. The example of that, this is Clostridium species that then grow under obligate anaerobe. In the thermophiles, we have Thermotoga that can grow, but in facultative anaerobes, we have Interbacter cloacae. The beauty of the Interbacter cloacae is that it can grow even in absence of oxygen or in the presence of oxygen. So it is easy to handle. And facultative anaerobe of thermophiles is the Klebsiella. Now, different substrate can be used for the biohydrogen production process. The carbohydrates, we have glucose, sucrose, maltose, cellulose, and cane molasses. Cellulosic material, we have energy crops like corn, wheat straw, paper pulp. Then waste, we have municipal solid waste, food waste, industrial waste, and the wastewater. And this is the metabolic pathway. If you look at complex organic material, when it hydrolyzes, they comprises of cellulose, hemicellulose, and starch. When they hydrolyze, they produce glucose, and this glucose undergoes this glycolytic pathway, what we call Emmett-Meyerhof pathway. It produces the pyruvic acid, and this pyruvic acid, when it formate with the help of formate hydrogen lyase, it produces carbon dioxide and hydrogen, and here hydrogenous, it produces that NADH. You can see that how it contributes for the hydrogen production. And also we have another products, we have ethanol, we have acetate, we have lactate, but only the problem is that if you look at the ethanol production is little bit undesirable because it consumes the NADH, and also lactate also consume that NADH. So if we can block this pathway, this NADH can be used for the for the hydrogen production. That is how we have carried out some experiment how we can redirect the metabolic pathway to produce to increase the hydrogen production. And this is the different bacteria we isolated in our lab. One is initially this is the Enterobacter cloacae, where this is identified by IIT Chandigarh, and then later on this bacteria identified at Klebsiella pneumonia because it has 99.8% similarity. Here they have 98% similarity as far as the 16S RNA concern. Then we have Citrobacter friendly, we isolated from high oil containing soil. We have Clostridium species isolated from the anaerobic digester, Clostridium acidobutylicum also isolated from the anaerobic digester, Bacillus coagulant we isolated from Suez sludge. And this is the thing that we develop in our lab. We find that I explained the biomethanation process, and I told you the biomethanation process comprises of two steps. One is the acidogenesis, another is methanogenesis. Now, the acidogens they convert the organic material to volatile fatty acid. And if you look at that methanogens, they convert the volatile fatty acid to methane and carbon dioxide. Now, here little information is available that on the how the potentiality on the this acidogens for the hydrogen production. We are one of the lab who reported that this acidogens has very good potential for the hydrogen production, and we are quite successful in producing that. And this is how this organism looks under the microscope. And another, as I told you, we use the thermophilic bacteria also to find out their suitability for hydrogen production. Now, this is the with respect to temperature, we can classify the organism in three different groups. One is thermophiles, another mesophiles, and psychophiles. Now, thermophiles, we have Thermotoga. I told you that different organisms we there. Clostridium species is there. Mesophiles, we have Clostridium, Enterobacter, Escherichia, and psychophiles, we have Geobacter species. Now, if you look at the stoichiometry of hydrogen production, is very interesting. The 1 mole of glucose can convert 2 moles of acetic acid and 4 moles of hydrogen if it follows the acetic acid pathway. But if it follows the butyric acid pathway, then it produces 2 moles of hydrogen. So, our intention is that how this organism can be more you can use the acetate pathway so that our hydrogen production can be increased to a great extent. Now, another interesting feature is that we have already shown that through the glycolytic pathway we get our organism produce pyruvate, and this pyruvate we get the acetyl CoA and formate, and this formate in presence of formate hydrogen lyase, it produces carbon dioxide and hydrogen. This is also present in our organism, and this helps for the hydrogen production. Now I am going to share some of our research finding. Here I want to point out that Ministry of New and Energy, Renewable Energy Sources, they made a hydrogen roadmap, a national hydrogen roadmap, and there they identified 3 different areas. One is hydrogen production, another is hydrogen storage, another is hydrogen utilization. So they have given because hydrogen can be produced through the chemical process, thermochemical process, it can be produced through the electrolysis process, it can be produced through the biological process. So this MNRE, they have given the responsibility to IIT Kharagpur to lead the research of biohydrogen in India, and we took 5 groups with us. One is IICT Hyderabad, another is JNTU Hyderabad, then Allahabad University, Banaras Hindu University, and TERI New Delhi. So we work together, we try to find out the potentiality of different organic waste suitable for hydrogen production. Finally, we came out with the 2 pilot plant, 10 cubic meter pilot plant. One we established at IIT Kharagpur, another established at IICT Hyderabad. So I want to share, because our major finding, one of the major finding is that it is very interesting that when we use any kind of waste material for the microbial degradation process, as you know that waste material is a waste for a particular industry, and it undergoes several treatment processes. So what happens actually, that during the treatment process the quality of the material is lost. So I told you at the beginning, for the growth of bacteria we required carbon source, we required nitrogen source, we required minerals, we required vitamin. So if we don't give all this material properly, then the organism cannot grow and multiply. So we add some kind of in this waste we add some kind of like yeast extract, we add, or malt extract we add, but these are the material are very costly material. So when we talk about the energy generation process, naturally we shall have to think for the substitution of this material, and we came out with some kind of suggestion that we find out how this can be material can be replaced by by some waste material, so that our process will be economically attractive. Now here we can show you that we use the different type of organic waste, and ultimately we came out with the conclusion that algal biomass, distillery effluent, de-oil cake, cane molasses and starchy wastewater is the most suitable raw materials for hydrogen production. Now industrial wastewater, we have distillery effluent, canteen wastewater we have starchy wastewater, industrial and organic residue we have cane molasses, de-oil cake and algal biomass. Now this selection of waste and residue depends on organic and carbohydrate contents, also the availability, suitability and the cost. These are the different factors that affects on the selection of the raw materials. Now here I want to show you that different type of de-oil cake we considered that as a substrate. One is groundnut de-oil cake, it looks like this. Coconut de-oil cake is looks like this. Mustard de-oil cake is looks like this. Sesame is de-oil cake is looks like this. Rice bran is looking like that, and soybean de-oil cake is looks like this. Now if we here our batch process and continuous process, we make a comparative study that Klebsiella pneumonia, when we use cane molasses as raw materials, we use the yeast extract, then our hydrogen production is 1.9 liter per liter of the fermentation broth. When you use the groundnut de-oil cake, this is we find 2.8. That means even yeast as compared to yeast extract, groundnut de-oil cake gives the better results. Now this is distilled dry grain soluble solids. This gives also better results. This is also waste material of the distillery industry. Then distillery effluent, we also use this material. We find that de-oil cake, groundnut de-oil cake is better as compared to yeast extract. So yeast extract is very costly material. We can, if replaced by that, then the process will be quite cheaper. The acidogenic, we try to, this is the pure organism, we try to replace with the mixed acidogenic mixed culture, where we can use the unsterile conditions, and using unsterile condition also we get better results in case of groundnut de-oil cake. Then Klebsiella pneumonia with cane molasses and groundnut de-oil cake with the 50 liter reactors, we get 2.9. This is all 2 liter reactors. And then thermophilic mixed culture, we use the starchy wastewater, we get 3.9 liter per liter of the fermentation broth. Then when you use cane molasses, we get 4.8. Distillery effluent is 2.9. So here we observe that the distillery effluent get the comparatively less hydrogen production as compared to cane molasses. But continuous system, we operated with Klebsiella pneumonia and acidogenic mixed culture and thermophilic mixed culture, and we find our production at the different level 20 liters with 3.3 liter per hour. Distillery effluent is quite less, 0.3 0.8 in case of acidogenic mixed culture, 3.38 liter per hour, and thermophilic mixed culture is 1.6 liter per hour. Now here we can this is the 20 liter reactor we operated. This is a custom design, custom made reactor. We design and fabricated locally, and we operated this fermenter. And by operating the fermenter, we get the hydrogen production about 1667.7 liter per hour. This is we use the immobilized column. I discussed the immobilized column has added the advantage as compared to this suspended cell culture, because immobilized column you can fix the cell on the solid matrix and pass the substrate, and it will be converted to hydrogen. So the cell mass leaking from the system will be very less. Now when you use the thermophilic organism, then this is the thermophilic dark fermentation process. We find cane molasses. This is the control fermenter, but previous one is custom made fermenter that we designed by ourselves. This is supplied by the New Brunswick, and we get 4.8 liter per liter of the fermentation broth in case of cane molasses, and distillery effluent, and 2.9 liter per liter fermentation broth. And COD removal is 60 to 70%. And this is the 10 cubic meter pilot plant that we operated. This is the 10 cubic meter reactor. This is the 500 liter. This is a 50 liter. This is 3 cubic meter reactor. You can see this is located here, and we operated this successfully. We prepared the seed culture here, and we feed preparation we do it here in 2000 liter reactor. We transferred in this reactor, and then finally we transfer into the 10 cubic meter reactor. And this cane molasses and groundnut de-oil cake we use 2.5%, and finally we reduced it to 1.5%, and we find that gas product hydrogen content is about 50 cubic meters in run 1, and the second run we got it 48 cubic meter. That is, if you volume by volume, you can say 4.8 volume per volume of the real fermentation broth. Now we publish two books. One is on biohydrogen production, fundamentals and technology advances. This is published by CRC Press. This is kind of textbook. If anybody interested to develop the knowledge on this biohydrogen production process, I refer this book. You should go through a book. I think you will learn that how this process can be operated. Another book we recently published. This is the first book on biohydrogen in the world. And biohydrogen means hydrogen followed by methane production. We find this process is sustainable, and this will be with us for the future. Now after this, I will operate a video on the operation of biohydrogen pilot plant, and anybody interested to get the information more on the process, they can visit our website www.bioh2iitkgp.in.
This video takes us on a virtual tour demonstrating the operation and the functioning of biohydrogen pilot plant at IIT Kharagpur. system for the 10 meter cube reactor. After the media is transferred to the 10 meter cube reactor, inoculation is done from the 500 liter reactor. Constant sampling of the 10 meter cube reactor is done to monitor parameters such as
Thank you.